in the final chapter of AMP1. Chapter 17 is about the special senses. You are already probably familiar with these senses. Taste, smell, vision, balance, and hearing are the special senses. To distinguish them from some of the other senses like touch, temperature sensors, pain sensors, things we've already been talking about. So what we'll see as we look at the special senses is we have much more elaborate receptors for these senses, and particularly kind of the organs surrounding these receptors are really meant to kind of block other stimuli and help these receptors hone in on their specific special sense. So in this video, I want to cover the first two, olfaction and gustation. So olfaction, you got it, is your sense of smell, right? And so this is located, as we probably already know just from experience, the sense of smell is really located in the nasal cavity. So what we're looking for, really, where that sense of smell comes from, is what we call the olfactory epithelium. We'll zoom in and look at that closer, but I want you to kind of notice where we are here in this nasal cavity. This olfactory epithelium lines kind of the roof of that nasal cavity. This is the cribriform plate, if you remember that from the skull. So we're kind of covering that. We're also covering the perpendicular plate of the ethmoid bone. We can't see that in this picture. And this olfactory epithelium is also on that superior nasal concha, if you recognize those structures. Let's look a little closer at what the epithelium actually looks like. Okay, so what we're dealing with, we'll spend most of our time looking at this olfactory epithelium. This is where the actual receptors are, but we're going to talk about this lamina propria as well. Right, so our sense of smell really comes from these highly, I shouldn't say highly specialized, but these specialized neurons, and they have their dendrites actually sticking out into this mucus layer on the olfactory epithelium. Now, each of these cells can have around 20 dendrites, and they talk about just this massive surface area that all those dendrites contain. In fact, your book says that if you looked at that surface area, it would actually be the same as the surface area of your entire body. That's how interested we are in gathering these smells or odorants that surround us. Now, these neurons, these sensory neurons, are going to get worn out. And so what we also see here in this epithelial layer are what are called basal cells. These are stem cells that divide and actually replace these neurons. This is one of the few examples in adults where neurons are actually replaced. So it could be interesting for some research there. Okay. Here we see, I did want to point, this mucus layer that the dendrites are suspended in is actually really important. These substances that you're going to smell actually have to dissolve in the liquid to actually be able to bind to receptors on those dendrites. Okay? The mucus comes from what's called an olfactory gland. So that takes us up here to this lamina propria. So between the epithelium and the actual skull bone, we have this layer called the lamina propria. This is going to be areolar connective tissue, blood vessels, nerves, and of course, these mucus glands or olfactory glands. They're of course producing these copious amounts of mucus that keep the respiratory epithelium moist at all times so that we can actually get those molecules dissolved. Right? This is what the sense of smell actually looks like. We're going to see that the sense of smell uses something called a G-coupled protein. This is an example of a second messenger system. When we were talking, one of the early chapters on the cell had different kind of receptors, and this is mentioned briefly, but we'll go into it a little bit more here, not super far. So notice what happens, that actual molecule, right, a chemical, whether that's from an orange or someone's perfume, it actually has to dissolve into this mucus layer in order to bind to this receptor protein on the surface. But this receptor protein actually just triggers kind of a chain of events. So notice what happens. We activate what's called a G protein. Don't worry about it, other than to know that it is part of this second messenger system where that initial receptor doesn't make the change or trigger that change, but it starts a series of events. So this active G protein is actually going to convert ATP, right, our cellular currency of energy, into a closely related molecule. Remember, this has 3 phosphates, trisphosphate. We're going to convert it into a monophosphate. We're going to take 2 of those phosphates off, and here we have what's called cyclic AMP. Cyclic, like a circle. Cyclic AMP is then who opens channels, right? So this is a little different. We talked about other ways to open channels with voltage or chemicals or pressure. In this case, we're using a second messenger, cyclic AMP, to bind to the channel and now open it, right? But once we open it, it's very similar. We're letting sodium in, and that is triggering the depolarization, and that is going to then lead to that action potential that our brain perceives, right? So a second messenger system, that's a little new one for us, okay? Now, notice what happens here. When we look at our sense of smell, we're actually picking up our stimulus with the dendrites of a neuron, right? So it's a modified neuron, essentially. And if we get enough of that stimulus, we'll cause a depolarization and send that message to the central nervous system. And so here they're just trying to point out, right, once we receive that stimulus, boom, 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 it keeps binding and binding and binding, we're able to send those action potentials. When we remove that stimulus, the rose in this case, right, we go back to our resting membrane potential. Now, a couple things to keep in mind here. Let's go back to this pathway, right? The way that action potential then gets to the brain, notice this, right? Here's the olfactory epithelium where those dendrites are embedded, right? They're actually going to then kind of bundle into these various little olfactory nerve fibers. Notice that's what's actually going through those pores in the cribriform plate. Remember seeing those little holes, especially if you looked at a real skull? Those are then all going to synapse here onto the olfactory bulb, which you hopefully saw in lab this week. That moves on to the olfactory tract, right? That bundle of axons in the central nervous system versus the peripheral nervous system, right? These were nerves. This is the tract. And that's going to then carry this to the brain. Now, the sense of smell goes to a few different places, right? We do take it to the cerebral cortex, so it can enter your conscious thought. But we know the sense of smell also goes to the limbic system, as well as the hypothalamus, and that's one of the reasons why there can be really strong emotions tied to certain smells, okay? Now, the last thing I want to say about olfaction, and then we'll move on, is olfaction is one of these senses that's like classically gets harder. How do I want to say this? You smell something that smells good, right? And then you very quickly seem to lose track of the fact that it smells like delicious Thanksgiving dinner, right? So we talked about this with our general receptors with this idea of adaptation. Now, I kind of lied to you, right, as we talked about tonic and phasic receptors, because olfaction, actually, the olfactory receptors don't do any adaptation. As long as the stimulus is there, they're sending that action potential. So how is it that you don't smell the turkey anymore? Well, in the sense of smell, we have multiple different synapses before we actually get to the cerebrum, right, to perceiving that smell. And it's actually the synapses along the way where this, they call it central adaptation, takes place, where we decide, nah, that's not such an important message to send anymore. So central adaptation is actually quite quick here with our sense of smell. Okay, let's talk a little bit about the sense of taste then. And we all know that the sense of smell and the sense of taste are actually quite connected, right? You taste things much better when you have your sense of smell. Have you noticed that before? Maybe a time when you had a cold or were sick. If you're really stuffed up, food kind of becomes bland, right? So these two senses are actually quite tied together. Now, the sense of taste, are you all familiar with this idea that we have major tastes, right? What are those? We have sweet, sour, salty, bitter, right? Those are the main four. And then your book also taught, so sweet, sour, bitter, salty. The book also talks about umami, which is like a rich flavor. They talk about it being like broth. And then also taste receptors for water. And that's an interesting one, right? Most of us would say water doesn't actually have a taste, but they point out when you take a nice long drink of water that you actually decrease the release of certain hormones. So clearly there are receptors and this message is being taken to the central nervous system. So we have all of these different tastes, and for the most part they're pretty evenly distributed. All of these different taste buds can pick up on these tastes. There's a little bit of difference maybe in different regions of the tongue. Okay, now a key thing to understand is that when you look at the surface of your tongue and you see all the bumps and ridges and things, those are not taste buds. Those are actually papilla, right? So notice we have all these different types of papilla, and the papilla actually harbor the taste buds down on the sides of the papilla. They're actually kind of protecting them, isolating the taste buds from that I don't want to say undigested, but like the food before it's really been kind of mashed and chewed and processed. Once it's been mashed up enough and dissolved in liquid, that liquid can actually move down the sides of any of these papilla and come in contact with the taste bud. And the taste bud then is this group of cells that actually have the receptors inside of them, right? So papilla and taste buds are different. Now, if we look at the 4 different types of papilla located on your tongue, these filiform papilla tend to be towards the front of your tongue, and they do not have any taste buds. They're really just like friction points, right? They're actually what's going to help you kind of move food around your mouth. I tend to think of these as the ones like if a cat were licking you and it kind of feels like sandpaper. Ours aren't as rough, but same idea. They do not participate in this sense of taste. They're more for actually mechanically moving the food around, right? Our other papilla here do have taste buds, right? So the fungiform papilla, we'll see kind of scattered around the tongue, and they have about 5 different taste buds on each papilla. The folate papilla are actually towards the back of your tongue. You're at home, so you can do this. Pull out your tongue, look in the mirror. You'll see these kind of ridges running down the side of your tongue. Those are the folate papilla, right? And so you can see the taste buds on the side there. And then the valate papilla, they're very large, and they make this V, so V, think valate, on the back of your tongue. And those can actually have hundreds of taste buds located down, again, on the sides of those valate papilla. Okay? So the valate papilla have the fewest bumps, but each of those papilla is actually really rich in those taste buds. Okay, so let's zoom in and look at what's going on here in this actual taste bud. So again, we're on the side of one of those papilla, right? And here we can actually see the little taste buds sticking out into the side. And so this is what it looks like. You have basically the actual receptor cells, what they're calling here a gustatory cell. And notice this cell, it is interfacing with a neuron, right? So it's actually separate from a neuron. It's a receptor cell. It's not like olfaction where the receptor was a neuron. Now we have a receptor cell that touches a neuron, right? And this receptor cell has what are called taste hairs. It's really just describing the microvilli on the surface. And then of course it's for surface area, but all these little microvilli are poking out through what's called the taste pore. So this taste bud, in order to actually touch the chemicals in our mouth, is putting microvilli out this little pore, right, where it can come in contact with them. But notice what else is in here. We also have basal cells. These are stem cells that are going to divide to keep replacing these gustatory cells, or taste receptors is fine, right? And these, so you see the basal cells, this transitional cell is like a teenage taste receptor. And so you kind of have multiple generations of those receptors going in there all the time. And the turnover is actually pretty quick on those as well. But again, when we think about that turnover, the turnover is not of the neuron like it is in smell, but just this receptor cell, specialized cell. Okay? Now this is what the sense of taste actually looks like. These work a little bit differently. So salt and sour are just sodium ions or hydrogen ions moving through a leak channel and depolarizing a membrane, right? And that kind of makes sense. The flavor, the taste is actually based on the presence of an ion. When we get to sweet, bitter, and umami, we're back to that G protein, that second messenger system. So we see the molecule actually binding to a receptor on the surface that activates this G protein, and it's actually that active second messenger. And here they're not telling us it might not be cyclic AMP, it might be a different second messenger, but it's actually that second messenger that's the one that's actually causing the stimulation of the neuron. So it's causing the release of those neurotransmitters that goes to the neuron. Because remember, we have two separate cells here, which means we actually have a synaptic delay, right? That receptor cell receives the stimulus, but then it actually needs to release a neurotransmitter to cross that gap. Right, here's our receptor cell, neurotransmitters are going to cross that synapse and then trigger depolarization of the actual neuron, right? So again, notice here, the stimulus is causing a bit of a depolarization. This is just going to be a graded potential, not an action potential. But then if it releases the neurotransmitters, right, then we can actually trigger this action potential in the neuron. The sense of taste is going to then travel through a couple different cranial nerves. It travels on the facial nerve, the glossopharyngeal nerve, and the vagus nerve. And what we're going to end up doing with that sense of taste, remember the insula, they're kind of separating that temporal, kind of parietal frontal lobe. If you dig in there, you get to another region of cortex called the insula, and that is the location where this information for taste goes. Also keep in mind, right, your sense of taste, we said, is very much linked to that sense of smell. So the brain is clearly kind of coordinating, perceiving things differently based on the input of both of these senses at the same time.